DNA, the second most important thing after the invention of the blue lace. It is this tiny thing inside us that often determines traits like personality, hair color, and even eye color. For most of history, scientists could not see the DNA, no matter how big their magnifiers were. So how did we go from squinting at these tiny little things to printing out billions of copies of them like receipts? The answer is this small thing called PCR, or like normal people would call it, the thing your nose suffered through in 2020. To understand where it all started, let's go back to the very beginning. Are we really doing this again?
Yeah, we are.
No. So before the invention of the PCR, scientists couldn't just check DNA like you check WhatsApp messages for typos, which is odd because microscopes were invented centuries ago. So why didn't they just zoom in?
So seeing the DNA was not the actual problem. In fact, 6th graders have experiments where they extract DNA from plants like strawberries and bananas. The real problem was detecting the molecules that make up the DNA. There are 4 molecules that we call nucleotides. That's adenine, guanine, thymine, and cytosine. And each DNA strand has billions of these. The difficulty arises when you are trying to detect a single mutation between these billions of nucleotides. So earlier genetic testing methods involved a complicated method called Southern blotting. So it involved cutting up the DNA, sorting them by size, and adding radioactive probes to detect these changes. And these were incredibly slow. It would take days to even weeks to detect a change.
Would they have detected the disease faster than I can cross the Silk Road Junction?
So let's take the example of cystic fibrosis, right? It's a single nucleotide mutation on chromosome 6 or 7. So before PCR, you had to cut up the DNA using extraction enzymes, run it on a gel so you can separate it by size, transfer it on a membrane, and add radioactive probes that only attaches if the mutation is present. And even then, the radioactivity would be very faint. It would be really hard to detect.
So you're telling me that to find one wrong letter, you had to slice someone's DNA into noodles, cook it in electricity, irradiate it, wash it, and hope it glowed? Sounds like a World War I recipe.
More or less.
And all this because you guys couldn't zoom in? Have you tried using a bigger microscope? Like a really big one? Try turning a telescope around.
That's not how microscope... never mind.
This is a PCR machine, the invention of which is credited to Carey Mullis. He was a Nobel laureate, a biochemist, a surfer, and a part-time mushroom eater. To know more about him, we have arranged an interview with a Mullis expert.
He was a brilliant scientist, but he was also unconventional.
Him again for
He was a brilliant scientist with a PhD from UC Berkeley, but he was also unconventional with his methods and known to be abusive and unpredictable with his co-workers. Basically, he was an He worked at a small biotech company called the Cetus Corporation, where he was tasked with making DNA probes. But one fine day, they bought in a machine which could do it automatically. So he was kind of out of a job and he had more time to work on his own endeavors.
Right. So how did a drug addict come up with the idea of stuffing people's noses with cotton swabs and win a Nobel Prize for it?
He actually did take many days off to relax. During one of these relaxing drives to his cabin in the mountains, he thought to himself, what if we amplify the DNA and make copies of it and try to detect the mutation in them? Because trying to detect one mutation in one copy of DNA is much harder than trying to detect that same mutation in a plethora of copies of DNA. The mutation also would be amplified, right? That's what he thought to himself. He said, and I quote, "I saw pink and blue molecules in between the mountains and my car. Now, he claims that he wasn't on LSD at this time, but he had done so much of it that his brain had just learned how to get that high without the drug itself.
So how exactly does PCR work? And make it short so that we don't have to put up a Subway Surfers video at the side of the screen.
So PCR has 3 steps, right? It's denaturation, where the 2 strands of DNA separate at 95 degrees Celsius, and then annealing, where 2 specially made primers attach to both the strands of DNA. And then we have extension, where a special enzyme called DNA polymerase attaches and then extends and fills in the gap between the strands. So this step is repeated again and again up to 30 times, where you get 1 billion copies of the required DNA. But the problem here was that the DNA polymerase used would melt at the high temperatures of denaturation at 95 degrees. So they had to come up with a way to make this automatic.
So after inventing the PCR, Kary Mullis went on to win the Nobel Prize. And he was also such a big druggie that he could feel the effects of the drugs without even taking them. So would you say psychedelics are the key to scientific breakthroughs?
That is true. Mullis does attribute his success to the use of psychedelics like LSD. That does not mean that everyone else should do that and they would also have success using those methods, right?
So could you look straight into the camera and say, kids, do psychedelics before you do your homework?
No, I'm not saying that.
Could you at least smile and nod like you agree? Close enough, we can fix that in post.
Kids do drugs.
But the problem was, the polymermates kept dying. A bit like my houseplants or every single goldfish that I've owned ever since I was 6. Early PCR was like boiling pasta but turning off the heat every 30 seconds. Very inefficient, but that was considered cutting-edge technology back in the day. You'd think that evolution would have considered humans to be boiling it repeatedly in a laboratory someday. So how exactly did they overcome the problem of not being able to boil the polymermate?
Nearly a decade before Mullis had his idea of PCR, Tom Brock and his student Hudson Fries had discovered a bacteria living in the hot springs of Yellowstone National Park, and they had named it Thermus aquaticus. It was thought to be useless at the time, but after PCR was invented, the enzymes used by that bacteria were very heat resistant as they lived in boiling temperatures. So their polymerase, called Taq polymerase, was used in PCR, which solved the problem of having to use the enzyme over and over for every step.
So you're telling me a guy named Fries discovered a bacterium in boiling water? That seems pretty ironic. Also, how did Mullis feel about getting all the glory for his work?
Well, it was a little complicated. His colleagues at CETUS wrote the paper and published it without naming him as an author, not because they did not want him to be there. They actually urged him to write a paper on his own first, but he procrastinated so much that the other companies were catching up and were in the process of filing patents for it. So they wanted to get their paper out first, so they had no other choice than to publish without him.
So exactly like a university assignment where one person does the work and the others get the credit?
Yeah, I suppose you could say that. Kary Mullis argued that he was the sole inventor of PCR and wanted full credit for it. He went on rants and he argued that he should get full credit. But at the end of the day, it was his own procrastination that led him to not get the credit for it. But the Nobel Committee did recognize his work. In 1993, they awarded him with the Nobel Prize for his fundamental contribution to the invention of PCR. Interestingly enough, his colleagues did not get it.
So in conclusion, the 2 ways of achieving a breakthrough in biology is either by accident or by doing copious amounts of drugs.
What? No, that's not... What?
So PCR did not just solve lab inconveniences. It changed everything, including those ancestry reports that tells you that you have a cousin in Africa. So the next time you're getting a PCR swab test done, you'll know that it was because of a man who got bored at work and got high. If I get caught by the police again, I'm just going to tell them that the drugs was because I'm achieving a scientific breakthrough and on the verge of getting a Nobel Prize.
Went on jail, transferred to a membrane.
No, not like that. Think of it like you're at a party, you're talking to a girl, she's not interested in biology, but you're just nerding out to her regardless.
Jokes on you, I have experience with that.